Hello, so I'm Hannah and I work down at the Dove Marine Lab in Cullercote, and we have this long-term time series that's been going on since 1968, and it's actually the UK's longest fixed-term station for plankton, and also benthic, that means seafloor animals as well. And so if you follow the Dove Time series on Instagram, it's where I put up lots of photos of the critters we find, so it's a little bit more interactive, and I give a bit of spiel about what they are and how they've adapted. And because I can talk quite a lot about this for days on end, I decided to make it a bit more interactive. So there'll be photos of all the critters we find. These are all photos I've taken from the species we get in the North Sea. So first one being, any ideas what this is here? If you get no answers, I will pick on random people. All right, Jack, do you know what this is?
A jellyfish.
No, it's actually a mollusk. It's called a sea angel, and it's related to slugs, and its shell's actually in the inside. And it's adapted its foot into a pair of wings and uses it to swim. So how do I go about doing this? So it's been going since 1968, 5.5 miles, nautical miles off the coast of Blythe. We actually have 2 benthic stations as well, but we're not talking about that today. And we have 3 different nets, small, medium and large, and I can actually fit in the biggest one, although it's designed to catch things that are only 5 mils or so in size. And we have 3 nets because we want to capture the whole range of plankton. And things will, the size of zooplankton, which is the animals, that will range from 0.2 millimeters up to barrel jellyfish. So it's from the smallest things to the largest. And there's 3 main approaches that I'm looking into. So I'm looking at how things change over time for this long-term monitoring. What's the community in the North Sea doing? Then looking at how can we look at biodiversity through time on historic sample collections, and then looking at molecular and morphological comparison and how to integrate this to get the most information we can for policy. So why zooplankton? It's actually a key role in aquatic ecosystems. So phytoplankton, which is all the algae, they actually produce 50% of our O2. So every second breath we take actually comes from phytoplankton. So without it, all life on Earth would cease to exist. So then the zooplankton will feed on that, moving energy up to higher trophic levels. And then is involved in nutrient cycling and actually carbon sequestration. So they move the CO2 from the phytoplankton, they eat it, and then when they either poop or die, that then sinks to the seafloor, and then all those critters get something to eat. So it's not all about this guy here. It's a very bad depiction of plankton. It's actually more like these guys. This is a Calanus fin martius, or in normal person, it's a copepod, and they're the favourite food source of fish, and particularly fish larvae. These guys are full of fat, and they're about 2 to 3 mils in size, and without these guys, fish larvae would have very little to eat. And then it can range zooplankton to something like these. These are 2 stages of a particular marine species. So it's the same species, just at 2 different larval stages. Any ideas what these are? Almost. The back end of the big one, this is a juvenile. It's on the way to a settlement stage. If you look at the bottom of it, it's all soft-bodied, curled up, so it would need an external, something else to find to put its back end into. You often find it in rock pools. Yeah, that's a hermit crab. So it starts off like this, like a shrimp, generic-looking shrimp, and then it'll turn into this guy here. And then he'll find his way to the seafloor, and then hopefully we'll find a shell to put his butt into. So also indicators for environmental health. So the zooplankton have a really rapid life cycle, often lasting a max a year in total, but they also have rapid reproductive cycles, which means any impact that we do, these react first. So anything we do, there's pollution, temperature changes, large amounts of fresh water going into the sea, these guys will react first. And then also they provide a lot of stability. The greater number of species means there's a greater stability for things higher up the food chain. And like here, this is just some of the species I found within the plankton. So you can see how diverse and how wide-ranging this is. So why do you want to do long-term monitoring? Why do you care? Why do you want to do it for a long period of time? So if you do it over decades, we can see the seasonal variations. So intra-annually, so within a year, you can see what's happening between winter and summer. And also the long-term population shifts. What's happening since the 60s to now? What's the ecosystem status right now? How is that compared to the 60s? Is it better than the 60s? It may not be great, but is it better? Or is it worse? And you can't detect changes or our impacts or the impacts of natural cycle until you establish a baseline. What is that normal? What's the normal that we expect to find? So the longer the time series, the greater the evidence we can have for management and policy in order to protect our fish stocks and the general health of the sea. So why do I want to integrate the standard stick under a microscope idea and count it with the molecular analysis? And I'm undertaking something called metabarcoding, which is where you take a sample, smush it all up, and then extract the DNA and sequence everything that's in it. So to stick something under a microscope takes a very long time to do. It requires a lot of funding, years worth of expertise. I mean, I've been doing it for 10 years and I know a tiny amount. Because there's 14,000 species of copepods, and there's the marine plankton is more numerous than all terrestrial invertebrates. So not that it's a competition, but marine wins in this one. And then what's the degree of similarity between institutions? How do you know that I've called something the same as somebody else? So you want that comparison between different institutions so you can compare what's going on all around the UK. And also, we want to find out what the community composition is, especially as a lot of larvae will spawn into the plankton, and they're so tiny, it's really hard to separate them. Then we also have things like gaps people have missed, and sometimes that things are damaged when you collect them, or something else has decided to munch on its back end, so you're missing half of it, which makes it impossible to ID. So out of these two images here, one of them is a larvae, and one of them stays in the plankton all its life. So any ideas what's on your left-hand side? Yeah, somebody said it over here.
Starfish.
Yeah, it's a baby starfish. And actually the blob next to it is how it starts out. They have various stages. They start out like this little blob with lots of like cilia, hairs, folds in it. And then it grows into this, what looks like a starfish, and that tail they use to move around and find a good place to settle. And also it gives something for fish larvae to eat rather than the actual self. So the fish larvae can eat the tail without actually damaging the species itself. So here is some of the stuff that looks pretty similar. So here we've got urchins, brittle stars, sea cucumbers and starfish. So they all look pretty similar, especially if you've got hundreds of them and they're all grouped together and they've been a bit damaged. Some fish decide to eat half of them. And then on the left, that's what happens to it if you've got, there's a CPR which the Plymouth uses to collect plankton, and this is basically a big metal container with a spool of silk, and as it passes through the water, these things get trapped on it. And that's what happens to it if it goes in a spool, so it makes it almost impossible to ID by just looking at it. So, any ideas what commercial species this is? Almost. Yeah, it's a langoustine. So, long-term changes in the plankton community. So, we want to know what the functional groups are, what they're doing. So those are the things that form a key role in the plankton, for a particular purpose, and things like that. And it's also good to picking up invasive species or non-natives. And actually, because where we are, it's the only one in the North Sea and the coast. Next closest ones are where the North Sea meets the English Channel. So actually it's highly important where we are. So I won't make you do these ones, I'll tell you what they are because they're quite difficult. So at the top is a worm larvae. It's actually called a shovel worm because its head looks like a shovel. Then we've got the three that look like jellyfish. There are species that are related to jellyfish. They're called hydroids, and they form these colonial groups that look a little bit like seaweed, but all spindly and spidery on rocks, and they actually spawn like this. So they're part of the Nidaria, which is jellyfish. Then this is a marine worm called Tomopteris. It doesn't actually have a common name apart from pelagic worm, which is very vague. And then this is just some of the stuff that in one sample. One snapshot, this is through a compound microscope, so I'm looking through the magnifier, and that's about 1 millimetre across. So you can see all the different things we get in just 1 millimetre of water. So over time, this is what's happening around the UK between 1960 and 2019. So when it says holoplankton, that's the stuff that stays in the plankton all its life. And where it says myriaplankton, that's the larvae of everything else. So you can see that the North Sea behaves very differently to the rest of the Atlantic. Although it's a sea, it's quite enclosed, it's quite impacted by a lot of human influence, and especially given the amount of ships that are going through the North Sea. But the downside is that we've got the larvae in the North Sea, it's going up. But the holoplankton is staying the same. But the larvae eat everything else. So if they're going up, it's not a sustainable trend. There's something that's going on that will not last forever. And at some point, the species that are always in the plankton will decrease, meaning there's less food for the larvae of other things. And the large copepods are the big juicy ones that the fish larvae like to eat, and they're actually declining, which means it has a massive impact to our fish stocks. So cod, haddock that people like to eat means they're running out of food. And everything is moving northward. As the sea warms up, everything is moving north. And we're not quite sure what's happening with the gelatinous zooplankton yet. That's all the jellyfish and things like that. Has anybody heard of the Portuguese man of war? Yeah, so that's actually a colonial species, so it's actually made up of several individual cells all together, and it's actually one of the most painful stings you'll ever experience if you ever encounter it. And actually that's moving northward. So it was a Mediterranean species, and that's moving to the UK. At the moment, it's just around London and the southwest, but with rising temperatures, there's a chance it might move up here. So if you ever see something that looks like an inflated plastic bag, just run away. So this is some of the stuff that we found. Total abundance of zooplankton in our samples from 2015 to 2022. And you can see around, just before the pandemic, it crashed. Not quite sure why that's happening yet, but it's probably something to do with the rising sea temperatures. So the langoustine, we have a big langoustine fishery in the northeast. It's really close to where my plankton station is. And this is what it's been doing for the last 5, 6, 7 years. So it has several peaks. They like to spawn in the summer. There's a lot of food going on. And you can see around 2021, it's a lot smaller than 2015 and 2022. Don't know why. Something bad happened in 2021 that affected the nephrops. It's either that the adults were overfished or something was happening to them in the plankton as a larval stage. Then you've got these dragonets. These are like fish that live on the sea floor and like to eat a lot of things that live on top of the sediment. And this is what their larvae looks like. And at the moment, it seems that their numbers are dropping off in more recent years, which is slightly worrying, because I think they're kind of cute, but also they're really good at keeping the sediment clean by eating whatever's on it. And then this one, any ideas? You eat it. Yeah, it's a lobster. But there you can see the impact of humans. So if you look at the tail on the left-hand image, you can see a blue thread. That's microplastic. That's a microfiber, probably from clothes that wrapped around its tail. So this guy was about to settle with plastic wrapped around his tail. So looking at biodiversity through time. So the Dove Time series is one of many large collections of biological samples. And often things are fixed in formaldehyde, which preserves the features so you can identify it. But the downside of that is that it forms these methylene cross-links between DNA and proteins, which means it's very hard to get the DNA out to identify what's in there, especially as things get older, the harder to tell they are as they degrade. Then also we put them out of the formaldehyde because it's a bit of a bad chemical, and then we put them in methylated spirits, which then fragments out the DNA further, which then has a negative impact for me to be able to extract the DNA. So testing extraction methods, well, there's a lot of help for me from people who have got more expertise, but we're testing a bunch of methods, modifying them, and it's found that it's worked on a couple of test samples, so we're going back and doing some more over the decades to see what we can get from there. So this guy, we also eat this guy, similar to the last one, but more round, has a pie crust on it. Yeah, it's an edible crab, and they're really easy to identify because they have the spine on the front and the spine on the back, whereas the others don't. So these are one of my favourite. So contemporary comparisons. So looking at samples that I've purposely taken for genetic analysis and morphology together, divide them in half. This thing here, this is all the plankton that we've got, and it's all red because that's all the fat that's in it, hence why the fish like to eat it. It's all the fat coming up. Split them in half using this thing called a Folsom splitter, and then they go into these little tubes here. That's for genetics, and then I take the other half and count it under a microscope. So I take a bulk sample for genetic analysis, not the water, because we don't know where that DNA has come from. Whereas if I take a bulk sample, I know that DNA has come from an actual thing. And then I've taken 30 samples and compared them. So one of the things we have to do is collect it, bring it to the lab, and then we get, because it's a whole year, and there's more in the summer as opposed to the winter. Which means there's varying quantities of essentially chunky plankton soup. So then we have to put these steel beads in and grind it to make the chunky soup into a more smoother soup to get the DNA out of. So there's 3 different species here. Any ideas what these three species are? I mean, it's easy for me, but I've been doing this for a while. Go on, what's the one in the middle? What's it look like? Yeah, that's an oyster. The one on this side here, that is it's a crab, but the first of its common name is the name of another terrestrial species. People are often scared of them if you find them in the bath. Yeah, so that's a spider crab. Then the one on the other end, it's a type of lobster, but not the one that people often eat. People do eat it, but they tend to eat the tail. We do very well in the gym, yeah. It's a squat lobster. It's actually monida is the Latin name. We have 2 species of squat lobster, glacier and monida. That's the fancy one. The other one's a bit boring, so I left them out. So we had varying quantities of DNA between the samples. That will be from winter to summer. It's going to be expected. And we took the CO1 gene. That's a gene that we like to use for plankton because it's quite common. And easy to extract. Then the molecular work found a species of non-native invasive copepod, this tortanus here, and actually it's becoming quite invasive around the world, especially around the UK. And then molecularly, crabs, worms, barnacles and bivalves were detected more via genetics and morphology. Often because bivalves, when they're that small, they just look like a bivalve until they get older. So looking at them can look like a bit intense, but looking at the different groups of taxa, the molecular will always get more species detected every time. So it's good to have that blended together. For that insight into biodiversity, what exactly our plankton community is doing. And that provides the accuracy and scope into biodiversity assessments. Therefore, that can feed into policy and decision makers. So if there is some sort of mitigation on our langoustine, they can have accurate information as to what's happening to it and what stage is something happening to it. Is it the adults? Is it the larvae? And at what point is it the larvae? Is it when it's newborn, or is it when it's settling? So outcomes of this, what influence, what's the point? So it's enhanced biodiversity. The more we know, the more we can do something about it. Time and cost effective. So the molecular analysis means that we can quickly identify what's in there, and then go into their morphology to look at the key areas, and then improve detection of environmental changes. As the zooplankton acts as our early warning sign, we want to know straight away, rather than having that delay onto fisheries, on the seafloor as well. So just as a slightly negative ending, so human beings largely are a little bit gross, and there's a study that we did a couple of years ago by someone before me, Priscilla, looking at the amount of microplastics in the plankton. And microplastics are plastics that are less than 5 millimeters inside even. And basically the plastics, they degrade into smaller and smaller parts. So plastic never biodegrades, it just gets smaller. And when it gets smaller, the surface of that gets more abrasive. And as it gets more abrasive, different things will stick to those plastics. That could be food, chemicals, waste, anything will stick to that. And they will ingest those microplastics and will build up into their tissues, called bioaccumulation. And fish actually have a higher uptake of these microplastics than crabs do. So anybody in this room eat fish? No. So if from 1971 to 2020, significant proportion of microplastics end up in our fish, and those fish make it to adulthood, and then we eat the fish, we're ingesting a hell of a lot of microplastics, which is not necessarily great. And alongside those microplastics are hazardous chemicals that are found within that. And the biggest one is actually cellophane, so all the plastic wrap that we have for food, that's one of the biggest problems. So don't ask me what you do about that. That's not my problem. I just tell you that we have an issue. So, any questions?